So welcome to this webinar for biology. In this series, we're doing one chapter at a time, and this webinar is going to cover all 4 sections of chapter 17 in the biology textbook. And we do have some practice questions on here that come up as polls. They are just for practice, they're not being graded, and no one can actually see your individual answers. I just see the group's answers as percentages. So please participate in all the quick poll questions. They are to review and make sure you understand it and let me know if I need to go back to anything. And they also just kind of keep your brain working so that you get to participate instead of just listening to me talking. And then you can definitely ask questions. The way to do that is to click on the question icon, which if you're looking at a computer is going to be this little speech bubble. And if you're looking at a phone or a tablet and you're on the app, it's going to be this question icon, and you just click on that and it'll let you type a question that comes to me. So once I see the questions, I'll answer them. I'll just keep an eye out for them as I go. You can ask questions about what I said. You could ask me to go back and go over something, or you could ask any other questions that you have about this topic. Okay, so don't hesitate to ask questions. You won't be interrupting anything at all. Okay, so this chapter is about the evolution of populations, so how populations change. So this chapter is about the way that populations change, and we need to remember that population is a group of organisms of the same species living in the same area. Let's review some other terms from previous chapters before we get going. Genes are pieces of DNA that code for a certain trait. For example, there are genes that control eye color. Alleles are versions of the genes. So there are specific alleles for blue eye color, brown eye color, etc. Each individual has a genotype and a genotype. The genotype is the set of alleles that the individual has for certain genes. If it inherited the same allele from both parents, it is homozygous for that gene. If it inherited a different alleles from its parents, it is heterozygous. For example, if there is an allele for brown eyes and an allele for blue eyes, they might be represented with a capital B and a lowercase b. The heterozygous genotype will be one of each. The homozygous dominant genotype will be 2 alleles for brown eyes, and homozygous recessive will be 2 alleles for blue eyes. Okay, so heterozygous means there are 2 different ones, and then homozygous means they're the same. So we either have 2 of these for brown eyes or 2 of these for blue eyes. Okay, the other term though is phenotype. The phenotype is the physical trait that is expressed. For example, the person has brown eyes. So phenotypes don't always completely match up with genotypes. If a person has this one, they're heterozygous. They could have a blue and a brown allele, but they're going to have brown eyes. Okay, so in some traits, there are dominant and recessive alleles. This means if the individual is heterozygous, they inherited one of each allele, the dominant allele will be expressed and the recessive allele will not be. Individuals can carry recessive alleles and pass them on to their offspring even if the individual does not physically appear to have them. So as we were saying on the last slide, if you have a dominant allele and a recessive allele, the recessive one is hidden. You're just going to have a phenotype showing that dominant one. But it still exists. You can still pass it on to your offspring. And that's what I just said. Okay, so they can still pass those individuals on to their offspring. The individuals of a population each have two alleles for all of their genes, one from their mother and one from their father. All of the alleles for all of the genes in all of the individuals in a population make up what's called the gene pool. In this lesson, we will be thinking a lot about allele frequency, which is how many times a specific allele occurs in the gene pool. Whenever evolution is occurring, the allele frequency of the population will change. That's really important to know. So if there's no evolution, allele frequency stays the same. If evolution is occurring, the allele frequency will change. In the above population, there are a total of 20 alleles for eye color, 12 alleles for blue eyes, and 8 for brown eyes. So the frequency for blue eyes in this example is 60%, which we write as 0.60. Okay, we write it as a decimal instead of a percent. And for brown eyes, it's 40%, written as 0.40. So why are all of the individuals in a population different? Well, there's a few sources of genetic variation. First of all, in sexual reproduction, the chromosomes inherited from each parent are a random combination. So brothers and sisters that have the same parents still have different combinations of alleles. During the process of meiosis, when the cells divide, and create the sex cells, crossing over can occur on some chromosomes, which means that the combination of genes on a chromosome will also be different. So even if you inherit a chromosome from your mother, it might be a little bit different than the one that she has because it's been crossed over. Okay. Mutations is another source of genetic variation. Mutations are accidental changes in an individual's DNA. That individual will have unique DNA, and sometimes this makes a change in the phenotype. And the third way that variation can occur is from what's called lateral gene transfer. This is a process that happens in single-celled organisms where they exchange genes with each other, and this gives them new combinations of genes. So when you put all of these three factors together, it keeps genetic variation. No two individuals in a population will have the exact same genes, always different combinations. So some traits are controlled by single genes, and they are aptly named single gene traits. Other traits are affected by several genes working together, and these are called polygenic traits. Polygenic traits are often traits that have a range. A good example is height. People don't come in simply a tall or short variety at set heights, but people come in a range of heights of all different sizes. If we graph the heights of humans, we get a bell curve. So a large percentage of people would have heights near the average, and a smaller percentage of people would have heights that are very tall or very short. So when we have polygenic traits, they tend to be like this, where we've got a whole range. Some people down here, most of the people in the middle around the average, and then some people on the opposite extreme. So before we go too much further, well, there's more text explaining what I just said. So most people are near the average, and then you have a few people on each side, much smaller or much taller. But before we go too much further, we want to review. So here's a review question, and I'm going to launch this as a poll. So our question is, if an organism has a dominant allele and a recessive allele for the same gene, they have what? Okay, so you should see that pop up now as a question on your screen for you to answer. So we need everybody to answer that so that we can move on. So think about it based on what we just heard. Okay, can we get that last person to vote? I'll give you another 10 seconds. Okay, I guess we'll just stick with what we've got. So we have half people said heterozygous genotype and half said heterozygous phenotype. So at least you got the idea that it's not homozygous. Okay, you have one of each, it's heterozygous. So let's go through our answers here. Homozygous genotype is not it. That would be two of the same alleles. So good job, nobody picked that one. Correct answer is heterozygous genotype. And so what's wrong with these last two, homozygous and heterozygous phenotype? Those terms actually don't really exist. Phenotypes are not described by heterozygous or homozygous because a phenotype is just the trait that you see. It doesn't matter what genes are behind it. So we might say the phenotype is that a person is blonde or a person has blue eyes or brown eyes, regardless of what their genes are. Okay, so our next section, 17.2. Another term to review is fitness. The higher an organism's fitness is, the better suited it is to living and reproducing within its ecosystem. Individuals with high fitness have traits that help them survive, and they pass their genes on to their offspring. This is called natural selection. In the next generation, the gene pool will contain a higher frequency of the alleles that help the organism be fit, and less of the alleles that do not, and the population will shift more with each generation. Okay, so an easy example of it, you have genetic variation. Some of the beetles are green, some of them are brown. If the birds want to eat the green ones, then they will. That makes it less fit to be green and more fit to be brown. They survive better if they're brown. So after many generations, the green ones get eaten, the brown ones survive, and the brown ones are the ones that reproduce. So the whole population shifts to being brown instead of green. Okay, so again, it's all about the individuals with high fitness are the ones that survive. And because of that, the population will shift with each generation. So this is how excuse me, I'm sorry. This is how natural selection works on single gene traits, where it's pretty straightforward. It's a little bit different with polygenic traits. Natural selection on polygenic traits is a bit more complicated. You've got three different things that could happen. Directional selection is similar to the process that we just described, so that the whole population shifts one way. So in these graphs, the red is the original population and the blue is after selection. Okay, so in this case, you had directional selection. It was better for the trait to be like this, so the whole population shifted that way. You could also have what's called stabilizing selection. So stabilizing selection occurs if the individual thrives when its traits are average. It's best not to be one extreme or the other. So our graph looks like this. It stays in the middle, and the forces of natural selection even force more of the individuals to be closer to the average. And our third option is a weird one. This is called disruptive selection. Disruptive selection is in the opposite, when either extreme increases the individual's fitness instead of the individuals in the middle. Okay, an example that was given in the book is birds on an island, where large beaks are useful for cracking large nuts, and small beaks are useful for getting small seeds out of tiny plants. And birds with average sized beaks can't really do either of those very well. So the population is going to change in two directions, either to have very large beaks or very small beaks, but no medium size. So that graph would look like this, where we start out, again, red was the original, and then blue is after selection. Nobody's in the middle, but you've got more individuals with a higher or lower than average. So these are all types of selection that happen on polygenic traits. Because remember, polygenic Those are the ones that are controlled by more than one gene. They're the ones that tend to give us a range like this where we graph it out. Okay, so single gene traits tend to be more on or off. Okay, or something very specific. It's red or it's blue. Where polygenic is something that gives us a whole range. Again, like people's heights. We don't just have people that are 5'3" and people that are 6'3". We have everything in between. There's people that are 5'4¾", the entire range. So that's what happens with polygenic traits, and that's why we get these three different ways that selection can happen where the graph is going to move around. Okay, we're going to review this before we move on. So which of the following happens on single gene traits? So let me start the poll for that one. Okay, this one's only got three options. So if you have a single gene trait, not a polygenic trait, do we have disruptive selection, stabilizing selection, or change of allele frequency? I'll give you about a minute to answer this. Okay, and we're still percentage-wise, we need one more person to vote. Can we get one last vote in there in the next 5 seconds? Okay, well, looks like we had people saying stabilizing selection and people saying change of allele frequency. So let's look at our options. Disruptive and stabilizing selection are ones that only happen for polygenic traits. So remember, stabilizing will kind of force everybody to be in the middle. That would be stabilizing. And disruptive selection would be where nobody's in the middle. The middle is not selected for, but instead it's going to move lower and higher. So that leaves change of allele frequency. That is the only way that selection happens with a single gene trait. So more or less of the individuals will have that allele. Okay, and we actually have another review question on this section before we move on. Disruptive selection for polygenetic traits results in what? So is disruptive selection when we get two different extreme phenotypes? No change in phenotypes? The phenotypes all shift in one direction? Or it shifts towards having traits favored by humans? Okay, I'll give you about 30 more seconds to vote on this one. Okay, I'm going to go back to this one because we got some wrong answers here. So, all right, polygenic traits. There's three different ways that you could have selection. You could have directional, you could have stabilizing, or you could have disruptive. So this question was about disruptive selection. So disruptive selection, you can think of kind of disrupting what's normal. It's going to change it up. So it's when it goes to either extreme. You've got high and you've got low, but not the average. It's going to decrease what's in the middle. So disruptive is going to look like this blue line, where it's decreasing the amount of individuals in the middle, and it's increasing the amount of individuals that have a really high or really low number for whatever trait it is. Now, this could be for anything, really. We talked about height. The book's example was birds' beaks and measuring the size of their beaks. So pretty much anything like that where it comes in a range where you would be measuring a number. So the example, or I'm sorry, yeah, this is example question. To disruptive selection for polygenic traits results in two different extreme phenotypes being created. So these three wrong answers, if there's not really any change, that's stabilizing, it's keeping it towards the average. If it all shifts in one direction, it's going to be directional selection. So either the whole population shifts to be bigger or smaller. And then we didn't really talk about this, but it's in the book. Artificial selection is having traits favored by humans. Okay. Also in this section is talking about genetic drift. So what we've been talking about so far is evolution that makes sense. There's a certain reason that the changes are happening. There's some sort of natural selection pressure that's making it happen. But when you have really small populations, they can shift randomly, and this is called genetic drift. Genetic drift can be caused by either the bottleneck effect or the founder effect. So we're going to look at both of those. The bottleneck effect is when there's a chance event, such as a flood or a disease, that causes most of the alleles to die, or most of the population to die, and the alleles of the individuals that happen to be left alive are different from the original gene pool. Okay, so here's our population of red and blue and green and yellow smileys, and an event happens, in this case a flood, that's going to get rid of most of the population, and randomly this is what happened to be left, was our yellow and green. So they're going to repopulate, and now our whole population is going to be yellow and green. There's no red and blue left. So this is what happens with the bottleneck effect. The populations will be rebuilt from those individuals that happen to survive, and they will have different traits and much less genetic diversity than the original population. Pretty similar is the founder effect. The founder effect occurs when a few individuals move into a new habitat that doesn't already have a population of that species, and then the new population in that area will be built from those individuals, which may have a different set of alleles than the original population's gene pool. This effect has been seen clearly when small groups of organisms wind up on an isolated island, but it can also happen in less drastic circumstances. When a population is not evolving but staying pretty much the same generation after generation after generation, this is called genetic equilibrium. There will still be differences between the individuals due to all the reasons we talked about before. There's always genetic variation in the population due to the shuffling of genes and sexual reproduction mostly. But the allele frequency doesn't change. Okay, and this is only when the population is not evolving. We have genetic equilibrium and the frequency doesn't change. And this is described by what's called the Hardy-Weinberg principle, after the two scientists that wrote it all out. This principle is that a population will stay in genetic equilibrium unless something causes the population to evolve. And here are 5 things that they would say would cause a population to change. Okay, non-random mating, where small population, movement into or out of the population, mutations, and natural selection. Okay, so non-random mating means that the individuals select other individuals to mate with based on certain traits, so the population will shift towards having those traits. a very small population can be changed more easily by random things like the genetic drift that we just saw than a large population. Movement of individuals from one population to another can change the allele frequency as they bring genes with them or take genes out. Mutations obviously create new alleles, and as we've discussed, natural selection, by definition, causes a population to shift to be more fit. Okay, so these are the 5 things that work against genetic equilibrium. So if any of these 5 are happening, we don't have genetic equilibrium. But if none of those 5 are happening and our population is really stable in genetic equilibrium, we can use the Hardy-Weinberg formula. So the Hardy-Weinberg principle can be used to predict the frequencies of genotypes in a population that is not evolving. And we see this on page 413 in your textbook. It gives this formula for finding the frequencies of genotypes in a population with 2 different alleles. The formula is p squared equals 2pq, p squared plus 2pq plus q squared equals 1. So what in the world does that mean? In this formula, the p and the q are 2 alleles of the same gene. Okay, so if we use our example from the very beginning, we have blue eyes and brown eyes. So p can be brown eyes, q can be blue eyes, or the genes for them. So you have 2 different alleles. And then using this formula, p squared is 2 p's. So p squared is the homozygous genotype, and 2pq is the heterozygous genotype. It's got a p and a q, and q squared, 2 q's, is the other homozygous genotype. Usually p is dominant and q is recessive. So in the next couple slides, I've got examples, and I try to keep them color-coded like this. So p squared is going to be, again, the dominant. Well, p is going to be the dominant allele. P squared is homozygous dominant, and that's going to be red. And heterozygous is going to be green, and the other homozygous is going to be purple. When you add all of these up, so you add up the percentage that is homozygous dominant and homozygous recessive and heterozygous, you get 1. So your numbers always have to add up to be 1. So on page 413, the first sample problem has A as the dominant allele and lowercase a as the recessive allele. In the first formula, P will represent the dominant allele and Q will represent the recessive allele. The question tells us that the frequency is 60% for capital A, so we'll replace all the P's in the formula with 0.6. Okay, because instead of percentages, we use decimals, so 60% becomes 0.6. So we're going to replace, every time there's a P in the formula, we replace it with 0.6. And the problem tells us that frequency of the recessive allele A, lowercase a, is 40%. That becomes 0.40. So we're going to replace all the Q's in the formula with 0.40. So P plus 2pq plus q squared equals 1 becomes 0.6 squared plus 2 times 0.6 times 0.4 plus 0.4 squared equals 1. Okay, so everything is just on the right here is exactly the same spot on the left. We had p squared, so we're going to replace the p with 0.6, and we get 0.6 squared. And then we had 2 times p times q. So now we have 2 times 0.6 times 0.4. Again, all we did was just replace the letter p with 0.6 and replace the letter q with 0.4. And then our last term on the right here, we had q squared, so that becomes 0.4 squared. So, now we just do a little bit of math. So p squared became 0.6 squared. And because it's squared, we just have to square it. 0.6 times 0.6 gives us 0.36. And now we can change it back to percentages and say 36%. Okay, so 36% of the population has this genotype of being homozygous dominant. Our next term was 2pq, and this represents the heterozygous allele because you have a p and a q. So we take 2 times 0.6 times 0.4, and that gives us 0.48. So 48% is heterozygous. And our last one is q squared, and that becomes 0.4 squared, and 0.4 times 0.4 is 16. So 16% of the population will be homozygous recessive. Okay, let's do one more example. Suppose a population has a gene with a dominant allele with a frequency of 30% and a recessive allele with a frequency of 70%. What is the frequency of each gene? So we're just going to put those numbers into this formula. So our formula is the same. P squared equals 2pq plus q squared equals 1. So we need to change our percentages into decimals. So 30% becomes 0.30, 70% becomes 0.70. And then those are the numbers that we put in the formula. So every time we see a P, we're going to replace it with 0.30. Every time we see a Q, we're going to replace it with 0.70. So the dominant allele is P and the recessive allele is Q. That's what it said. Oops, I'm sorry. And so we just replace them. So P squared becomes 0.30 squared. And 2 times P times Q becomes 2 times 0.3 times 0.7. And Q squared becomes 0.7 squared. So all we did was take these numbers for the frequency, okay, and use those to replace the P's and replace the Q's. Now, you just have to do the math. So 0.30 squared becomes 0.09, and 2 times 0.3 times 0.7 becomes 0.42, and 0.7 squared becomes 0.49, and that's where we can actually stop. If you want to check your work, you would add those three numbers up and find out, yes, they do equal 1, so that's good. We want our three numbers to equal 1. But with these questions, we just kind of want to know what's the frequency of each of those options. So the left, the p squared, remember that's homozygous dominant, so we came up with 0.09, and 0.09 is 9%. So 0.09 is 9% of the population will be homozygous dominant. And in the middle, we have 0.42. So 42% of the population will be homozygous, or heterozygous, I'm sorry. Okay, a capital and a lowercase, heterozygous. And 0.49, so 49% of the population will be homozygous recessive. Okay, so we have two review questions on the Hardy-Weinberg formula just because it's so important and also so tricky. So I wanted to review it before we moved on. So remember, Hardy-Weinberg means it's the formula for what's happening when the allele frequency doesn't change. So what populations does the Hardy-Weinberg principle apply to? So I'll give you about 1 minute to answer this poll before we move on to the next one. And we're only at 50%. I'm going to give you another 10 seconds for trying to get that last answer in. What do you think? Okay, well, for those of you that did answer, great job. Populations that are not evolving. So again, Hardy-Weinberg only works when the allele frequency doesn't change. So it's got to be populations that are not evolving. And we saw there was 5 different reasons it won't work, and this was one of them. So the mates have to be randomly chosen. If they're not, if mating individuals are looking for something specific, the population will shift that way. And also, it's not often something that works with very small populations because they can have genetic drift. So this is the right answer, populations that are not evolving. And one more poll. So what would the Hardy-Weinberg equation look like for a population in which the dominant allele, capital R, has a frequency of 24%, and the recessive allele has a frequency of 76%. Okay, so if you still are looking at this formula and it's freaking you out, don't worry, it'll be okay. You can use your book for it on the test, you can use your notes that have the formula, but you just need to know what numbers go where. So I'll give you about a minute or less to vote for this one. Uh-oh, is everybody afraid to vote for this one? I'm going to write the formula at the bottom here. So that's what our formula looks like without any numbers in it. So what does it look like once we put those numbers in it? I'll give you another 10 seconds or so to think about it. All right, well, okay, I'll show you. So our formula is p squared equals 2pq plus q squared. So which one's right here? Well, we can say this one's wrong because what's going on in the middle? We just have a number 2. So that's not useful. We need a 2 times p times q squared. So that's not right. This bottom one, we didn't square anything. We just have 24 and there's no square. And we have 76 and there's no square. So it's really important to remember it's not p plus 2pq plus q, it's p squared plus 2p squared plus q squared. So is this last one in the middle correct? We have our dominant allele is 24%, which is going to be 0.24. So wherever there's a p, we replace it with 0.24. So we have 0.24 squared plus 2 times 0.24 times 0.76, which is the other frequency, plus 0.76 squared. Whoop. Okay, so yes, this is the right answer here, this second one. Okay, so I would recommend if you're not sure about it, you need to go back and reread that. You can watch the part where I explain it on the video because you'll get a recording of this session. Or just YouTube Hardy-Weinberg formula and see if you can find another video where they animate it. But we need to move on to the next section, which is 17.3. And We've looked so far at examples of populations shifting, but when a population changes to express different traits more frequently, the individuals of that population haven't necessarily started a new species. Individuals from another population could show up and breed with them and mix up the gene pool. So to form a new species, reproductive isolation must occur. Reproductive isolation means that the individuals of the different populations can no longer mate and produce offspring. So again, we talked about things shifting certain traits. Maybe they are changing to a different color. Maybe they're getting taller or shorter, or the birds are getting bigger or smaller beaks. But they haven't created new species. So to be a new species, you have to have reproductive isolation, meaning that the individuals of that population can no longer mate with the other ones from a different population and produce offspring. They've created a brand new species. So for reproductive isolation to happen, the populations need to be isolated long enough to evolve significantly. There are a few different ways this can happen. Geographic isolation is when a physical barrier separates the populations. It could be a river or a mountain range or a glacier, anything that they can't cross to find mates. Behavioral isolation is when the behavior of the organisms keep them from mating. Maybe they are active at different times of day, or they develop different mating rituals. And temporal isolation is when populations reproduce at different times. Maybe one population mates in March and the other mates in June. All of these can isolate a population enough to develop a new species. And we don't really have time to go into it now, but pages 415 to 416 specifically describe how those different mechanisms worked to create evolution of new species of finches on the Galapagos Islands. And the last section in this chapter is 17.4, which is about molecular evolution. So studying molecular evolution means studying changes to the macromolecules in an organism instead of looking at the individual organism as a whole. So specifically, we focus on DNA. The DNA changes as organisms evolve. So by comparing DNA, we can theorize how closely different species are related. So one interesting way they do this is a molecular clock. So a molecular clock uses known mutation rates to estimate how long ago two organisms shared a common ancestor. They do this because some genes tend to mutate slowly, where the gene is passed down through many generations without being mutated, and other genes tend to mutate more quickly, meaning there are fewer generations between mutations. And if they know this about different genes, they can figure it out. So for example, if this is a tree of different species that are related, you can tell, obviously, that these two are more closely related than this one is to either of them, because look at the mutations. These two have two mutations that are the same, and this one over here has four completely different mutations. They're not the same. But when we're talking about a molecular clock, we need to know the mutation rates. And what they can do is figure out, okay, looking at these genes here that have mutated, there was only 2 mutations in this one, and now there are 4 mutations in this one. So by knowing the mutation rate of that gene and about how frequently it mutates, they can figure out how many years there were between this species and this species. Okay, was it 1 million years? Was it 20 million years? Was it 60 million years? They could figure that out based on the mutation rates of the genes. And then they could look at this and look at what genes mutated and figure out if they evolved earlier or later. So it's basically just figuring out how long ago the two organisms shared a common ancestor based on the known mutation rate. So that's called molecular clock. So another part of DNA, of course, is looking at the genes and which genes we have, and you'll see that different organisms have different amounts of genes. So how did we get so many? Probably we have so many genes from slowly building up genes through gene duplication. So how this works is originally a chromosome will occasionally end up with two copies of the same gene. So in my diagram here, we've got these 4 different genes on this chromosome, and now we end up with duplicate. So we have 2 copies of the same gene on one chromosome. That gives the individual organism an extra copy. So they still have one working copy to rely on that's not going to change, and then an extra copy that can mutate without affecting the working copy of the gene. So this new copy is free to mutate and change, and it can evolve. It can develop a new function while the original one is still there doing the original job. Okay, so that's gene duplication. And basically it's the process of a new copy of a gene evolving to fill a different role. And one of the last things they talk about is Hox genes. So how does DNA control the development of an organism's body? It does it through Hox genes. These are genes that control where body parts grow, how they grow, when they grow, and what shape they take. An example that they use in your book is the UBX gene. So this is a gene that's found in both shrimp and in insects. And the version of the gene in shrimp allows it to have many legs. But a mutated version of the UBX gene is in an insect ancestor has been passed down to all insects. And that limited the insects to having 6 legs. So that's just, So that's just one example, but there's many examples of Hox genes which control the growth of body parts. Okay, and that actually takes us to the end of the section. So we have one more poll for you to answer, and then one more review question that's not a poll, it's just on the screen. Okay, so molecular clocks use known mutation rates in certain genes to do what? Give you about a minute to answer this poll. All right, great job. So that's our answer is estimate how long ago organisms shared a common ancestor. That's what molecular clocks do with known mutation rates. And our last one, this is not one that I could make as a poll for you to answer because it's a matching question, but we talked about isolating mechanisms. So we're going to match the 3 of them up with their definitions. So we've got populations that are separated by a barrier. What kind of isolation is that? Populations that live in the same habitat but reproduce at different times. And populations that live in the same habitat but have different behaviors that prevent mating. So the first one, separated by a barrier, is geographic isolation. The second one, living in the same habitat but reproducing at different times, is called temporal. And the third one is different behaviors that prevent mating would be behavioral isolation. Okay, so that is all that I had to talk about in the chapter. If you are watching this live, I'm going to stick around for a few minutes to answer any more questions that you have. If you're watching a recording of it, this is how you can contact your instructor by email or through phone.